Hey guys, this is Dr. Mobeen. We're talking about central nervous system. Today's lecture is really exciting, and that is about the development of the brain, or the embryology of the brain. We will actually have 3 or 4 lectures that are related to this topic. This is the first lecture within the series that would be the brain development. The second lecture will be the spinal cord development. Third one will be the abnormalities or pathologies of the development, which are really important, and finally the neurohistology. Combined, I think we will cover about 10 questions that can appear in the USMLE. So please pay attention. The other thing for the rest of the students who are not doing the USMLE, this is a high yield lecture, more towards the USMLE concepts compared to just general embryology. So if you are looking for too many details for the embryology, this may not be the right lecture for you. However, we will explain enough that you can have a decent review and you can understand how the brain develops. So let's start. So the embryology of the brain, or the brain development, this lecture will not include the spinal cord development, and it will not include the pathologies. We will do that in a separate lecture. So let's start with this one. The first thing to remember is this. Usually the brain development starts in the 3rd week, and by 4th or 5th week the major structures have actually formed. So if there is a problem, for example, if the mother is alcohol abuser, or the substance abuser, or she is unexpectedly, unknowingly exposed to hazardous material, and many times girls do not know that they are pregnant, and within the very first few weeks the brain development occurs. So there is a lot of chance that the tissue damage can occur. So please remember, 3rd week and normally 4th to 5th week, a majority of the structures have come together. You would actually see that then till the 1st year of life, even after the birth, till the 1st year of life, there are myelinations and some developments that keep on going. And the second thing is, till the adolescence, the neuronal connections or synapses or interconnections keep forming. So major structure will appear from 3rd week to 4th or 5th week, but then development would continue. With this in mind, and I'll show you a table separately where we'll go day by day to say what kind of structure is coming when. Let us first understand what are the basic concepts of embryology or development of the brain, and then we'll look at the days. So first of all, let's orient ourselves. The zygote is formed, the zygote has developing cells in it, and, the sperm and the egg, they came together, they fused, they formed the zygote, and now the zygote is developing. What happens is that the very first stage is the Blastula stage, or bilaminar germ disc. How does that look? So let's do this. So this is the bilaminar germ disc, or there are 2 layers of cells that are present. Now these 2 layers, imagine if you take a ball, a sphere, and cut it in half and put one half above and the other half below. So I'm going to do that. I'm going to put a half above and I'm going to put another half below. So this is the initial structure. In this structure, here are how these are named. This is the primitive amniotic cavity. This is the yolk sac. This is hypoblast, and this here is epiblast. I named them wrong. This is epiblast and this is hypoblast. So these are the structures. Now the epiblast and the hypoblast are the 2 germ layers. This is the amniotic fluid. At this time in this embryo, the concept of the rest of the structures. Let me just take this out, it would look correct this way. In this embryo, the concept of anterior portion, posterior portion, cranial, caudal, apexial, hypaxial, these concepts are not there yet. These orientations are not there yet. So at the 3rd week, there would appear a primitive streak. As soon as the primitive streak appears, that would start creating these kind of the directional structure within the embryo. So let's look at what happens. Okay. So I've opened up the 2 spheres, the amniotic sac and the yolk sac. I'm not making the complete structure here. Now in the epiblast, what happens is primitive node and a primitive streak appears. As soon as this primitive node and streak appear, there are certain things that we can now think about. Number one, this area, the primitive streak, appears in the caudal portion. So now the embryo, developing embryo, has a caudal portion. The other side is going to be cranial. So cranial, caudal has become apparent. Second thing is, where the primitive streak is, the 2 sides, left and right, or 2 lateral sides, are now going to develop bilaterally, or you know that the structure that we have a median, and then around the median we are bilateral structure. We have 2 eyes, 2 ears, and we have bilateral symmetry in that structure. So this neural tube, this primitive streak and primitive node or Hansen's node start creating the bilateral symmetry as well. It divides the embryo, and now both sides would develop in bilateral symmetry. So what do we have here? We have primitive streak, which is this, and we have primitive node, which is this. This is also called Hansen's node. Now, almost at the same time, there is one more thing, and that is that the embryo develops a membranous area it will start developing a membranous area in the cranial part and one in the caudal part. So I'm just going to make that here. One is this and the other one is this. These 2 are the start of mouth and anus. So as soon as one can see the start of mouth and anus, of course the anal side will be the caudal side and the mouth side would be the cranial side. So now we've gotten the bilateral symmetry, plus we've gotten the caudal and cranial structure. So we've gotten the precaudal plate or buccopharyngeal membrane, that is this, and we've gotten cloacal plate or anal membrane, that is this. Now this all is happening in the epiblast. One thing that you would see as we progress this embryology further, you will see a 3rd layer to appear between the epiblast and the hypoblast. However, keep in mind that the precaudal plate and the cloacal plates, they would stay membranous, they would not thicken up. So the 3 layers, so let's say these are the 3 layers. This is the epiblast, this is the hypoblast. The 3rd layer is going to appear between them, which is going to be the mesoderm. However, in the area of the precaudal plate, mesoderm will not develop, and epiblast and hypoblast would fuse with each other to form a thin membrane. That membrane in future would then dissolve and open up, and that would become mouth. Same for the anus. So that is the structure here. Now let's continue. What happens after that? So next, what will happen is you'll see 5 cells. We call them bottle cells. Bottle cells will originate from epiblast and go through the streak and node and between these 2 layers. So I'm going to make that over here. So again, if you see in this one, what we did was, one, I separated these 2 layers a little bit. Why did I separate it? I want to make a new layer between them. So we created a little distance between them. Ideally, when the 3rd layer, there were the 2 layers. Let's say this was, I'm going to pick up, let's say this. So let's say there were 2 papers, and these were the epiblast and hypoblast. Between them, the fibers would come and start making a 3rd layer. So the distance would appear as the cells migrate. In my diagram, the distance has appeared before the cell come down, so apologies for that. Ideally, as the cells start migrating, the distance would start appearing. Now the question is, where are the cells going to come from? The cells are going to come from the epiblast. This is the future ectoderm as well. This is the future layer that would give rise to skin. So the cells for the nervous system and for the mesoderm are actually going to originate from the cells that would make the skin in the future. So what happens is, there are cells that are going to start migrating through the primitive node and primitive streak, and they would start coming down. So let's make some cells here. What are these cells? These are the cells coming down from these. So let's make them in red color. Cool. So these cells that are coming down, they are going to lay a new layer between the epiblast and the hypoblast. This will become the mesoderm. As soon as you get the 3rd layer, the names of the layers are now going to be changed. We are going to call them ectoderm, mesoderm, and endoderm. So let me make that over here, the 3 layers. Now one thing to keep in mind, the buccopharyngeal membrane and the cloacal membrane, or precordial plate and the cloacal plate, they would stay fused with the hypoderm. The fibers, the cells of the mesoderm will not come and make a layer there. So let's see that. Okay, so now what we're seeing is this. The cells that are coming down, they would start forming a tube. This is the notochord. This is the future area. So this is notochord. Notochord is going to be very, very significant, very important in the development. Why? Number one, this is the area that would create the axis. This is the area around which the vertebrae would develop. You'll see that the notochord itself, the remnants of the notochord would become nucleus pulposus of the vertebral disc. This notochord would actually induce, this is very important, this is a USMLE question, what structure would induce the formation of the central nervous system and the mesoderm? Notochord secretes inducing factors that would start the central nervous system development, and that would start the mesoderm around it to start appearing. So if the notochord does not form, or if the notochord does not start in providing the secretory substances, then the whole further development would fall apart. Now notochord is connected with what? The primitive streak and the primitive node, because the fibers were coming down, the cells were coming down from these places and were migrating to form this notochord. Now if I take one more slice here and kind of cut it like this and then see this slice. Let's see how that would look like. I'm going to make these three structures now. Okay, so let's orient ourselves to this coronal section. What is happening here is I have cut it here, passing through the primitive streak area, and we are now seeing from the front. What we are seeing is this is the, what are these cells? This is the epiblast, right? This is the epiblast layer. In the epiblast layer, this is the primitive streak. Of course, if we are seeing from this side, you would see the cloacal membrane somewhere over here. We cannot see the buccopharyngeal side because we have cut that off. Now this is the epiblast. Of course, this one, the lower one, is the hypoblast. Right? These are the hypoblast cells. Between the epiblast and the hypoblast, the cells that migrated from above, the bottle cells that came down, these cells have formed, number one, this structure, which is called the notochord. It's a tubular structure in the center between the two layers. So if there were two layers, if I pick up these papers, so if there were two layers, between the two layers, a tubular structure has appeared. This is the notochord. Now the notochord, and if you see more red cells around this, these are also the cells that are migrating from above, and you would see that we will make more structures with those cells. So this is the structure now. We've gotten the notochord. That is the important piece to remember. Once we've gotten the notochord, this is the notochord. I'm going to make this in black in all the next diagrams. This is notochord. Notochord would start neurulation, the process of neurulation. So first we went from blastula. Blastula is bilaminar germ disc to the gastrula phase, where we started developing the trilaminar germ disc. So you're seeing this is the start of the trilaminar. It's not a fully developed trilaminar germ disc. Notochord would now start the process of neurulation. Neurulation process is that it would induce central nervous system production. As part of that, the third layer would start developing here as well, and a gastrula would form. So let's do that here. Let's see what happens when the notochord start inducing. What will happen is, keep an eye on this epiblast. Here is what happens. The epiblast, I'm going to make it look like slipper now. As it is developing, the cranial region develops faster and broader. The caudal region develops slower. Okay, so now here we've gotten the primitive streak and primitive node here, cloacal membrane here, buccopharyngeal membrane here, and this area has started becoming bigger. What happens is, in here, a thickening appears, or the cells become thick. So now remember, this is still the epiblast, but the cells in the epiblast have become thick under the influence of the notochord, under the inductive influence of the notochord. The cell which are becoming thick, this thick region is called neural plate. Neural plate. So underneath, what have we gotten underneath? We've gotten the notochord sitting here, right? So like this. This is the notochord underneath, and then below that is the hypoblast and the yolk sac and the amniotic cavity. Let's do a slice of this as well to see how does that look. This thing, if we cut it from here, we are going to see this thickening of the cells or formation of this new plate inside the epiblast. So, these are the epiblast cells. Underneath is sitting the notochord, and below that sits the hypoblast. So, hypo is here, epi is here, and epi has developed this thickening in it, which is the neural plate. This is being induced by the notochord. Now, what happens is the next step for the neural plate is this. It is developing in the ectoderm, it is developing in the epiblast, but the central nervous system is not sticking out on the skin. What would happen is the thickness would finally sink down, and it would start making the spinal cord in the brain. And the problem is this: if the thickness sinks down, that would leave, if this is a paper, if I cut a piece of it and sink it down here, then the paper would have a hole. So, we need to somehow fill that hole as well. So, what happens is, as the thickness appears here, the sides of the thickness would come and fuse with each other, a tube will form, that tube will separate out, that will become the neural tube, it would come above the notochord. So, notochord has induced the epiblast above, that is going to create a thickening, then the margins of the thickening are going to lift up and fuse with each other, that would keep the epiblast as an intact layer. But if you see here, when the margins are going to lift up and fuse, there will be an area. So, normally the fusion starts in the cervical region. Once this fusion starts, there is going to be an area that is going to be open above, an area that would be open below. Then the fusion, like a zipper, will move upwards and downwards and close the whole structure, separating the tube out. But till that time, I want you to know this is clinically very important, USMLE, this is very important. Till that time, the area that is open above and the area that is open below is the neuropores. Why this is important? As this neural tube would start making CSF, the CSF can actually spill out of these. So, let us talk about that for a second. So, now what is going to happen is, this thickening is going to start invaginating. So, here is what happens. So, now what we are doing is, we are seeing the surface of the epiblast, we are seeing that this groove has started appearing, or a tubular structure which is opened, a tubular structure has started appearing and the cells have started sinking. In this structure, keep in mind, so let us first make the full structure here. This is the notochord right underneath. Remember, it was here, this was inducing the area, and that caused this thickening of the neural plate. The neural plate is now sinking and it is creating a fold which is called the neural fold. Now, in this neural fold, so this is the neural fold. In this neural fold, these cells here near the top are called the neural crest cells. They are making the crest of the fold, not the whole fold. So, of course, there is a floor of the fold and then there is a crest of the fold. The cells that are at the crest, just keep in mind that they are going to separate out and they would make a separate structure, and the fold, remaining fold, would close into a tube, and that would be a separate structure. Meanwhile, these are going to fuse as well. So, let me just make the little hypoblast layer here. This was notochord. Notochord seems like it is fused into the hypoblast. That is not the case. Notochord is above, between the epiblast and the hypoblast. Right? So, this is the notochord. So, we have gotten the neural fold, we have gotten the neural crest. Neural crest. Now, what will happen? Now, what will happen is that the lips of the fold or the crests, they would come together and they will fuse with each other. So, I am going to make that over here. Okay, so now what has happened is that these crests, they have come together and they have started fusing like a zipper. But as they fuse cranially on this side, we are cutting it like this, and caudally on this side. So primitive streak is somewhere over here, the cloacal membrane is somewhere over here. So now caudally this is going to start fusing, but meanwhile this is important. Clinically it is important, USMLE. This is a neuropore that has formed. A neuropore has formed. What is a neuropore? That is where the tube is not yet fused. Now where is the neuropore opening? It is opening upwards. What is above? What is above is the amniotic cavity. This is amniotic cavity, right? This was epiblast or the ectoderm. So this neuropore is opening into the ectoderm. Why is that important? That is important because of following thing. In the test in the USMLE and in clinical scenarios, they'll ask you what will happen if you find that amniotic fluid or the serum of the pregnant mother has gotten increased alpha fetoproteins. Alpha fetoproteins are part of the CSF. CSF is going to be inside the neural tube, which is going to become the brain in the future. However, in the beginning, the neural pores open it in the amniotic cavity. If those neuropores do not close, then the CSF producing will spill out in the amniotic cavity. From there it would actually appear in maternal CSF as well, maternal serum as well. So what would happen if a baby, developing baby, does not have the neuropores closed, what will happen to the AFP? These will be raised. The raised AFP in the amniotic cavity, amniotic space, or raised AFP in the maternal serum is an indicator that the neuropores may not have closed correctly. In that case, doctors can actually go do CT scans, ultrasound, whatever. They can even do intrauterine surgery as well to try to fix these things. Similarly, acetylcholine esterase is part of the CSF, and that would also spill out. So this is very important from clinical point of view and from USMLE point of view. So the neuropore, if I make the elongated embryo up here or down here maybe, I want to show two neuropores. So if I make this complete epiblast layer, it would look like this. This layer has gotten primitive streak and that cloacal membrane, buccopharyngeal membrane. Here was this thickening or the neural plate, which is now folding upwards. So the fold is now closing, and there is one opening on one side, and then there is one opening on the cranial side. The rest of the fold has been closed, and the cells are emerging. So you can say, think about a tube that is connected like this, and it is sinking down, it is coming down. Underneath that is the notochord, and underneath that is the hypoblast. So this tube, the neural tube, is forming, and it has two neuropores, cranial and caudal. The cranial neuropore is belonging to the head side. So if there is a problem with the cranial neuropore, for example, it does not complete, it does not close, then anencephaly would occur, brain will not form. Similarly, the caudal neuropore, if that does not close, spina bifida would form. So these are very, very important. And how do you know that they are open? You would see increase in AFP and acetylcholine esterase present in the amniotic fluid and maternal serum. That is an indication that the neuropores are open. Okay, let's continue developing here. So now it's very simple and very fast. What we need to do is we need to make the tube. So I'm going to make that this view here. So now if you see here, what has happened is that the neural plate has sunk downwards between the epiblast and the hypoblast. It is becoming a tube because the folds came together, the crests came together, neural crest, and started fusing. Now, if I make the neural crest here in green, these, they have started fusing, and their fusion has closed the epiblast. So the back, the skin, the area is closing except the neural pore, which would also close. Now what we need to do is this. In the next diagram, you would see that the neural tube would separate out completely, and the neural crest cell would separate out separately and separated from the epiblast. So they would separate from the neural tube and they would separate from the epiblast and create a separate structure. So let's see that. That is again very important from clinical point of view. Okay, so now what has happened? This is really important. Number one, this neural tube has separated out. Number two, these green neural crest cells have separated out as the neural crest cells. You'll see that they'll make sympathetic ganglion and they'll make the enterochromaffin cells and the C cells of the thyroid and others. So we will talk about them in a second. This is a neuropore. So this tube, the neural tube is continue here with the pore. Ultimately, this pore would close as well. This is the primitive striatal node, and this is the cloacal membrane. This is not a cord. Now, one more thing which I have been deliberately not showing is that around the notochord, mesenchymal cells, the cells that were coming down from the ectoderm, the cells that had been coming down here, these cells around the notochord would start developing into mesoderm. So if I make them here, let me make them here. What is this structure? Number one, we have a trilaminar germ disc. This is a trilaminar. I'm just going to write here, trilaminar germ disc. This is a gastrula, trilaminar germ disc. It has three layers: ectoderm, mesoderm, and endoderm. Mesoderm, if you see here carefully, on each side it makes three structures. There is the medial mesoderm, then there is intermediate mesoderm, and there is lateral mesoderm. The medial mesoderm is what is going to start making somites. Somites are what are going to make the muscles and the dermatomes, and they would make the cartilages, and sclerotome would make the connective tissue and stuff. So that is this area. The intermediate mesoderm or mesenchyme is going to start making the gonads and the renal ridges. So the kidneys and gonads will form from there, and then the lateral mesoderm would have the parietal and visceral layers, and we'll see about that, plus the lateral somatic walls will start coming in together. Again, this is not about how the structure, rest of the structure forms, but this is important to keep in mind. Why? Because in the development of the brain, you would see that the pharyngeal area, pharyngeal pouches, and the somites present in the cranial space of the mesoderm, if there are abnormalities of those, you'll see those abnormalities like Röthke's pouch or the craniopharyngiomas or the problem with the the thyroid cyst and so on. But remember, this is the medial mesoderm layer. You're also seeing that the neural tube has formed. So now if you think about it, this is an embryo. If you stand it up, you would see this is the back side of the embryo. This is inner side of the embryo. This is the spinal cord. This is the notochord, which would become, with the mesoderm, it would become the nucleus pulposus, and the vertebra would develop behind this. So here will be the vertebra, spinal cord behind that. Now let's see what happens finally when the structures close. We'll end up having almost the same thing, a little bit more cleaner here. Now what would happen? Let's take the neural tube out of here, and let's remove the epiblast or ectoderm from here and see what is happening in this area. This is how it is going to look like. Okay, So if you see here, this is the neural tube. I've removed the ectoderm, I've removed the skin from the back, I've removed this. So what are you seeing? You're seeing the neural tube. This is that neural tube. On the sides of the neural tube, you're seeing neural crest cells. On the side of the neural crest, you're seeing these long structures in the form of somites, or there are swellings present in the mesoderm, right? So this is the medial mesoderm with its swellings. Every swelling is a somite. Every somite would have a sclerotome and a dermatome and a myotome. Sclerotome would make the connective tissues, cartilages, bones. Dermatome would make the skin area, epidermis or the skin area actually, and then the myotome would make the muscles. And every somite would receive a nerve from this developing neural crest cells here and developing neural tube. So that is how there is one-to-one nerve and the muscle and the skin and the connective tissue supply. This is how that is developing. It would start developing from the cervical region, then it goes up and it goes down. Many times, if the neural pore above the cranial side, if that doesn't close, then the anencephaly occur and the brain does not develop. And if the caudal neuropore that does not close at the lumbar 4-5 level, then there would be spina bifida that would be seen. And there are types. There is spina bifida occulta, then there is spina cystica as well. We'll talk about that in the pathology of this. So this is the structure here. Now let's see this neural tube. Let's say it is all decent, it is closed properly, the ectoderm above this is all closed as well. Let's see what happens to the neural tube. How does this make brain and spinal cord? So I'm going to make that here. I'm going to take this, actually let me take this portion out. So remember, here is the neural plate, there will be neural, so here that neural tube, this neural tube, I'm going to pick it up and put it here. Okay, so this is the neural tube. Neural tube of course has the neural crest cells around this, neural crest cells. It also has in front of it will be the notochord, and then the other structures. Mesoderm is around this, ectoderm is behind it, and the endoderm is front. So what I have done is I've taken up this structure and it is showing up like this. I've stood up that embryo. So now the embryo is standing. What we are interested in is to see what happens with the neural tube on the cranial side. This is the cranial side, this is the caudal side. So cranial, caudal, neural tube, and neural crest cells and CSS. So now let's see what happens is, this is again, now these are your Semele questions. What would happen is 3 dilatations would appear on the cranial side of the neural tube. So the 3 dilatations are these are called prosencephalon, prosen. Pros is the prefix, encephalon is the brain, encephalon. Then meson, pro is the beginning, early, meson, mes is the middle, encephalon, brain. Then is the rhomb, encephalon. Rhomb actually means rhombus like structure because of the 4th ventricle and that structure that forms, we call it rhombencephalon. Now what happens is, after these 3 dilatations are formed, these dilatations are 2 of them. So this dilatation, mesencephalon, would stay like this. Mesencephalon would stay mesencephalon and will make midbrain. The prosencephalon and rhombencephalon would further divide into 2 more dilatation each. So if I make that here, there is going to be So, this is the mesencephalon. So, now what we have gotten telencephalon. So, prosencephalon has become divided into tel and di. So, telencephalon, this is telencephalon, this is diencephalon. Mesencephalon stays the same. Now, the rhombencephalon also divides into two. One is metencephalon and the other one is myencephalon. So, metencephalon is here and myencephalon is here. So, I remember it with this mnemonic: tell Diane me met my spinal cord. Spinal cord. Tell Diane me, which is mesencephalon here, me met my spinal cord. Now, what happens here is telencephalon would develop the fastest and it would develop the cerebrum. Now, important thing here, this is very, very simple actually. To remember this now, if you remember the ventricles, you can actually remember how this whole structure would form. What will this structure form? Every structure here has its own cavity, and whatever is around that cavity is the structure that comes from it. For example, telencephalon is going to be lateral ventricle. So, instead of trying to remember these names and then saying what are the cavities, remember it the other way around. Go with the cavities. Lateral ventricles, so if I make them here, lateral ventricle, third ventricle, aqueduct of Sylvius, then fourth ventricle, right? Then spinal cords, central canal. Lateral ventricle, third ventricle, aqueduct of Sylvius, aqueduct, fourth ventricle, and spinal cord, spinal canal, right? Think about it for a second. What is around the spinal central canal? Spinal cord. So, spinal cord is associated with this central canal cavity. Fourth ventricle, what is around this? We've gotten medulla in front, make it in red to make it clear, medulla in front and pons and medulla, behind cerebellum. So, all of these structures are around the fourth ventricle, they are coming from the things that would give rise to the fourth one, meten and myelencephalon. So, we will talk about it in a second. Aqueduct, what is around the aqueduct? The tectal part and tegmental part of the midbrain. Cool. So, the midbrain comes from the mesencephalon and the cavity it has is the aqueduct. Third ventricle, what is around the third ventricle? Thalami, hypothalamus, thalamus, subthalamus, epithalamus, these are present around the third ventricle. So, third ventricle, thalami, where would it come from? Diencephalon. So, thalami here. And then lateral ventricle, what is around the lateral ventricles? The cerebral hemispheres and the basal ganglia are associated with the lateral ventricle. So, that comes from the telencephalon. So, lateral ventricle comes from the telencephalon. Diencephalon is the third ventricle. Mesencephalon is aqueduct of Sylvius. Metencephalon and myelencephalon together, fourth ventricle. And then spinal cord, spinal canal or central canal, right? Now, what happens is the lateral ventricles, the telencephalon develops faster, and at the 40th day of development, there is the bendings that start appearing, so that the neural tubes start jamming into the cranial cavity, and all the structure has to get into the cranial cavity. So, the cranial cavity, you would start seeing some bends in the neural tube, and these bends are to accommodate the brain to develop within the cranial cavity. So, 40th day is when those bends start happening. So, telencephalon would give rise to the cerebral hemispheres and the basal nuclei around it, and the cavity is the lateral ventricle. Diencephalon would give rise to the thalami, the thalamus, hypothalamus and epithalamus are from the diencephalon, and what is the cavity between the thalami? Third ventricle. Mesencephalon, midbrain, this is the only one which does not further differentiate, it would stay midbrain. So, what are the parts of it? Cerebral peduncle, tectal part and tegmental part of the midbrain, these are all mesencephalon in origin, and aqueduct of Sylvius is their canal. Metencephalon and myelencephalon, these would give rise, meten would give rise to pons and cerebellum. So pons, if this is pons and this is cerebellum, pons and cerebellum together come from met, and medulla comes from myelencephalon. And then of course there is the spinal cord. These are the structures that are derived from the vesicles of the neural tube. Of course, if the cranial neuropore does not close, these structures would not start forming and anencephaly would appear, in which case there is no brain present. Again, if any neuropore is open, the neuropore opening can be detected by increased AFP and increased acetylcholine esterase in the amniotic cavity. The other thing, this is important, keep in mind, cranial neuropore, 25th day, 25th is very early in the development, 25th day, cranial neuropore closes, cranial neuropore closes. 27th day, 2 days after day, caudal neuropore closes. So if AFP and acetylcholine esterase are increased after the 27th days, then there is a suspicion that there may be a problem with the development. Normally what happens is, before the girl becomes pregnant, the folate treatment can help make sure that the neuropores would close. Normally before is better, if not then at least as soon as somebody finds out that they're pregnant, folate administration should start. That helps somehow closing the neuropores. Cool. So this is the development of the brain. We will now go to the next lecture, which will be the development of the spinal cord and the spinal nerves. Thank you very much, and we'll do the pathology after that. Thank you.